in this way, you will develop antibiotic resistance. Similar happens with water treatment. If water treatment is not effective, it stimulates accumulation of antibiotic-resistant bacteria in tap water. We can analyze which exactly antibiotics this bacteria resistant to by analysis of metagenome sequences. For example, from tap water, we have multiple efflux pumps, making bacteria resistant to various antimicrobials. In some samples from REVA, we can see aminoglycoside resistance genes associated with contamination of water with these antibiotics. So we can predict which antibiotics contaminate water in these specific regions. Another important way of analysis is transcriptomics. In this case, we want to use sequences to analyze how genes are used by bacteria, not only which genes are present, but whether bacteria use this gene and at which conditions. For example, here, it's interesting analysis where we analyzed distribution patterns of gene regulation in different Staphylococcus aureus microorganisms. Every dot corresponds to one sample, specific pattern of gene expression at the given samples, and we have samples normal conditions treated with one antibiotic, another antimicrobial compound, and by both of them. And you can see that we can tell difference between bacteria. At the beginning, we expected to see a grouping of dots depending on conditions, but in fact, it's not true. Different bacteria having very much similar genomes, similar genes, may use them in very different way. And this is very stable patterns of gene regulation and gene expression patterns. And one new paper was published last week. Basically, it's not yet available online. It's +1, where we make such general biological conclusion about clonal gene expression stability, which refer to the expression patterns of core and accessory genes characterized by clonal specificity and significant stability, regardless of applied stresses. We found also that the response to stress was also strain-specific, consistent with clonal gene expression stability. So when we're talking about personalized medicine, we have to consider not only that different patients have different organisms and require different treatment, but different bacteria having very much similar genomes, very much similar patterns of genes may respond differently to the same treatment with antibiotics because of this clonal gene expression stability. And another application is my preferable area of research for last decade is epigenetics. We can also analyze not only sequences of bacteria, but we can analyze which nucleotides are modified in these sequences, and these epigenetic modifications play a very important role in life of bacteria and in regulation of genes. First of all, methylation of bacterial genome is associated with restriction modification systems. This is immunity of bacteria. Methylases methylate bacterial genome at specific locations, at specific motifs. Then restrictase start to work, but it can't cut DNA at these locations because they are protected by methylation. If phages or virus or plasmids get inside of bacteria, they are not protected, and restrictases cut this DNA and protect bacteria from phages. But there are many other often methyltransferases which also methylate DNA, and these methyltransferases are important for regulation of genes. We developed new program and submitted paper now. Program is available online, and this program allows us to visualize distribution of methylated nucleotides on chromosomes and to perform statistics of which genes may be affected by this specific epigenetic modifications. Program is available online, but also is available at GitHub, so it can be downloaded and run on your computers locally. This program can be used to visualize specificity of pattern of distribution of methylated proteins, which specific motifs are methylated. What we found out that in the same staph aureus, which we used before, methylation patterns are quite different because methyltransferases methylated different patterns. And as I'll show later, it can explain different patterns of gene regulation. Also, the program analyze, perform some statistics, how this methylation is distributed, how it's biased to coding, non-coding regions or to promoter regions. In several organisms, we find a strong bias to coding regions, in other to promoter regions, which shows that this specific methylation may participate in gene regulation. And also we can tell difference by using methylation patterns between contigs from generated from metagenomes when we try to create metagenome assembled genomes. Very often we get hybrid, chimeric sequences because contigs come from closely related by different organisms. And by using this approach, we can tell difference between contigs which have the same pattern of methylation from contigs with different pattern of methylation and select only parts of genomes belonging to the same organism. Another interesting discovery published in this new paper is that methylation indeed can interfere with gene regulation. And we found specific regions in promoter regions where methylation of nucleotides may affect level of expression of genes, downstream genes. So it's not at every place the effect will be, we will find any effect on gene expression. Methylated sites must be located at very specific locations relatively to start codon position. In this case, its effect will be very strong, negative or positive effect on level of expression of downstream genes. So now we can use this approach to predict which genes are likely are down regulated. It's even not regulation, it's the level of expression. Which genes more likely will be overexpressed in these microorganisms, which genes will show low level of expression by using epigenetic profiling. There is another study, we compared Mycobacterium tuberculosis, and it was unexpected for me, it's very new results that these very conserved organisms differ significantly by patterns of methylation or different motifs. And these differences affect antibiotic resistance. In addition to mutations, which can be found in Mycobacterium tuberculosis, there is another mechanisms of antibiotic resistant development, is alternative methylation of whole genomes. And another interesting application of epigenetic studies is not only methylation. Many other nucleotides can get chemically transformed or modified. For example, guanine can be oxidized to 8-oxoguanine. And this oxidation is very important because it's increase level of mutations. So bacteria control the number of oxidized guanine residues, but if bacteria is under stress, it can't control it properly. On the left side, you can see distribution in green of modified adenine, and in brown, it's a modified guanine, oxidized guanine. In normal conditions, guanine drop quickly. There is a small number of oxidized guanine. But then we applied one new antimicrobial treatment. And you can see immediately, we apply concentration not enough to kill bacteria, but we can now analyze what happens with this bacteria. And you can see a strong increase of oxidized guanine as a response to this treatment. So we can measure how effective is new antibiotic by measuring of a level of oxidized guanine in bacteria. And on another study, we applied this to analyze for metagenomic study. We use PacBio sequencing for metagenomics. Then we identified frequency of oxidized guanine at different bacteria extracted from soil and at different conditions. This is soil rhizosphere from wheat plant, wheat field, taken in August. In South Africa, it's winter, it's rainy, rather cold. Then it was beginning of October, and weather was very good. And then in November, it's already drought condition, very hot and dry conditions. And you can see amount frequency of guanine residues in different organisms. Some bacteria suffer from cold and rainy conditions. For others, these conditions are good, but they suffer from droughts. In red, it's shown increase of guanine, oxidized guanine residues. So we can apply this technology to predict specifically which bacteria in metagenomes suffer from stress conditions and for which bacteria these conditions are appropriate. Why it's important? For example, if you want to introduce new beneficial bacteria to protect plants, we have to find such conditions where this bacteria optimal for this bacteria. Okay, so a few conclusions. I think my time is out. Those new opportunities arise with advent of novel sequencing technologies. However, there is many challenges, which includes the need to process large volume of data in every evolving formats. New software tools continue to emerge, but keeping up with rapid advancements remains difficult. And collaboration among programmers, bioinformaticians, and web lab practitioners is key to overcome these challenges. I want to thank my Center for Bioinformatics and National Research Foundation of South Africa for continuous support. Thank you, and I'm open for your questions.
Okay, thank you, Oleg, very much for your presentation. And I believe we have time for a couple of questions. We have one question in the chat from Julia.
Okay, I can see it's a very good question in chat about RNA. How are we to identify modifications RNA with sequencing? Great question, because only now we are able, with development of new nanopore technology, for the very first time we are able to sequence RNA directly without converting RNA to cDNA. And so now, and this work started already, there is zero knowledge about epigenetic modifications on the level of RNA, also modifications in DNA or RNA of viruses or phages. But we know it's play a very important role, and basically there is most likely much more mechanisms are involved. There is already new publications are published, but very general. We don't know in details what happens in the field. So whatever we find, it will be novelty. So it's a very good field for future studies.
Thank you, Oleg.